Dear students, hope you'll be fine. Today we are going to discuss very important topic, that is the DNA replication, or we can simply say it as replication of DNA. Now, before going into detail this, what do you mean by the DNA replication? अगर हम DNA की बात करेंगे on the basis of functions, DNA अगर हम on the basis अगर हम बात करेंगे फंक्शंस के एतबार से, डीएनए इट परफॉर्म्स टू इंपॉर्टेंट फंक्शंस. फर्स्ट वन, that is called as autocatalytic, and second one is called as heterocatalytic. DNA, it performs two important functions. First one is autocatalytic function, second one is heterocatalytic function. Now, what happens in the autocatalytic function? What happens there, जो DNA होता है, it directs the synthesis of DNA itself. Synthesis of DNA itself. So in case of autolytic, what we will see, this DNA, it directs the synthesis of DNA itself. That means that it forms the photostate copy, or we can say duplicate copy of itself. That जो इसकी photostate copy बन जाती है, जो इसकी duplicate copy बन जाते हैं, that auto means what we call as a self, वो itself बन जाती है. There we will see in this autocatalytic that it directs the synthesis of DNA itself. So we can say that the synthesis of DNA, it takes itself, that is जो इसकी कॉपी बन जाती है, जो इसकी डुप्लीकेट कॉपी बन जाए, जो इसकी फोटोस्टेट कॉपी बन जाते हैं, that is formed itself. And the second method जो हमारे पास है, that is called as this heterocatalytic process. Here we will see in autocatalytic DNA खुद ब खुद बन जाता है, synthesis of DNA itself. लेकिन यहाँ पर क्या हो जाता है कि DNA जो है, it directs the formation of what we call as the formation of chemicals. Directs the formation of chemicals other than itself to synthesize, या यहाँ पर हम क्या देखते हैं कि formation of other chemicals, what we will say other than. यहाँ पर हम क्या देते हैं, DNA खुद और कोई DNA नहीं बनाता है, लेकिन यहाँ पर हम क्या देते हैं, different chemicals बन जाते हैं from this DNA. So यहाँ पर हम क्या कहते हैं कि DNA जो है, it forms, here we see the formation of other chemicals takes place. लेकिन heterocatalytic जो हमारे पास है, इसमें और कोई डीएनए नहीं बनता है, लेकिन यहाँ पर हम क्या देखते हैं कि फॉर्मेशन ऑफ न्यू केमिकल्स बन जाते हैं. फॉर एग्जांपल अगर हम देखेंगे, here we see the synthesis of RNA takes place, here we will see the proteins takes place like this. So the new substances that are formed, they are not the substances which are formed, the chemicals they are formed, they are not the DNA, but there are different substances which are formed from the DNA, like RNA are formed, like the proteins are formed. Lekin in case of the autocatalytic, here we will see the synthesis of DNA itself takes place. Now, we have nothing to do with this heterocatalytic in the DNA replication, but we have to discuss this autocatalytic function, how the synthesis of DNA takes place. Yeah, we can simply say how this replication of DNA takes place. Now, जो replication है DNA की, जो mechanism है, जो process है, whatever the mechanism of this DNA replication is, it may take place. इसको हम आसान बनाने के लिए कि that this whole mechanism of DNA replication, it can be studied under various processes, under various steps. So the whole process of DNA replication, it can be discussed, it can be studied under various steps, and these steps, they require the use of more than dozen enzymes. So यहाँ पर हम क्या देखते हैं कि whatever the reactions, whatever the mechanism takes place, that whole process of replication या duplication of this DNA, it takes place with the help of enzyme. Now the first step, अगर हम वो बात कर लेंगे, what is the first step in the process of this DNA? First step, what we'll see, that is the first step, activation of, the first step is activation of deoxyribonucleotides. The first step is in the process of the DNA replication is activation of deoxyribonucleotides. Now if we go into detail, अगर हम पुराना याद दिलाएंगे, that there are 4 types of nucleotides in DNA. There are the 4 nucleotides of DNA. There are 4 different types of DNA. First one is AMP. This is the first nucleotide. Second one is हमारे पास CMP. थर्ड हमारे पास जो न्यूक्लियोटाइड है दैट इज GMP, एंड जो हमारे पास लास्ट न्यूक्लियोटाइड इज दैट इज TMP. एडेनोसिन मोनोफॉस्फेट, साइटोसिन मोनोफॉस्फेट, guanine monophosphate, and TMP, thymine monophosphate. So we have got 4 different types of nucleotides: AMP, CMP, GMP, and TMP. And what we will see, that these 4 types of these nucleotides, they are seen floating in the nucleus. They are seen floating in the nucleus. Thus, in order to activate these nucleotides, what we do, they are being activated by ATP. To activate this AMP, to activate this CMP, to activate this GMP, to activate this TMP, what do we need? We need the ATP, adenosine triphosphate, in order to form the deoxyribonucleoside. So these nucleotides, they are being activated with the help of these ATP. First, they are found seen floating in the nucleus, then they are being activated by this ATP in order to form the nucleoside triphosphates. This is the first step, and these are called as ATP, then we will see GTP, then we are called as this CTP, then we have got TTP. Now in this first step, we need an enzyme in order to activate this. These nucleotides, they are seen floating in the nucleus, and to activate them, they are being activated by this ATP to form the deoxyribonucleoside triphosphates, these are called as ATP, GTP, CTP, TTP. And for these, the enzyme required, इस स्टेप में हमारे पास एक एंजाइम का इस्तेमाल होता है, and that enzyme is called as phosphorylase. And that enzyme is known as phosphorylase. And this first step, it is also called as, because of this presence of this enzyme, It is called as phosphorylation. So first step in this DNA replication is activation of deoxyribonucleotides. We have got DNA has got 4 different types of nucleotides, and these nucleotides are seen floating in the nucleus, and these nucleotides, they are being activated by APT to form the deoxyribonucleoside triphosphate called as ATP, GTP, CTP, TTP, and the enzyme in this required at this step, that is called as phosphorylase, and this step is called as phosphorylation. This is the first step in this DNA replication. Now the second step, which we are going to discuss, that is recognition of initiation point. Recognition of the initiation point. Now कि अब हमको वो पॉइंट देखना है on the DNA molecule from which this replication can take place. कि वो पॉइंट देखना है on the DNA from which this replication of DNA takes place, and that step is called as recognition of initiation point. हाँ, what we will see, जहां से ये DNA replication start हो जाती है, that is called as initiation point. That is the starting point of replication, starting point for replication. That is the starting point for replication. Now to identify this initiation point, we need on the DNA molecule, we need some proteins, and these proteins are called as initiation proteins are needed. To find this, to what we call as to locate this initiation point, to identify this initiation point, we need some proteins. These proteins are called as initiation proteins. These initiation proteins, they help us in locating this initiation point on the DNA. Now in case of prokaryotes like virus, like bacteria, what we will see, there may be only one origin, there may be only one this initiation point for replication. In prokaryotes like viruses, like bacteria, here we will see there is only one point of replication, while as in case of eukaryotes, what we see, जो वहां पर हमारे पास DNA molecule जो होता है, that is very much large. In case of eukaryotes, वहां पर हमारे पास क्या होता है, in eukaryotes वहां पर हम क्या देखते हैं कि DNA molecule is somewhat large, वो बहुत ही बड़ा होता है, and in this we will see there is not only one initiation point, but there may be many initiation points, there may be many origin points from those points, from those places, the process of replication can take place, and all these points, they finally merge, वो आपस में मिल जाते हैं, and they merge with one another. In eukaryotes, what we will see, DNA molecule is very much large, and there may be many initiation points, there may be many initiation points, there may be many initiation places from which this replication can take place, and all these points, they finally merge with each other. So first step हमने क्या पढ़ा, that is the activation of deoxyribonucleotide, and second step is the recognition of the initiation point. Now in the third, we have got 2 more steps. 2-3 steps more, and those steps we will try to discuss in the another topic, that is the unwinding of the DNA molecule. Second, that is fourth point हमारे पास आता है formation of this RNA primer, and last one is the formation of the new DNA chain. We will discuss them in the other topic. Until then, goodbye. God bless you. Thanks.